Good afternoon, everyone. So today's topic is fetal neurodevelopment after maternal anesthesia. The goal behind this topic is to take you through the literature on anesthetic neurotoxicity and try to help you understand how relevant it will be to your clinical practice. So before I begin, I'd like to disclose that I have no conflicts of interest. So the first question is, why should we be concerned? Well, there's a lot of literature out there since the year 2003 showing that administration of anesthetic agents, both intravenous and inhalational, cause extensive neurodegeneration. And this appears to be an age-dependent phenomenon and happens especially during the phase of synaptogenesis, which in humans usually extends from the third trimester to up to the third year of life. So the first study in this topic was published by Dr. Vesna Jevtovic-Todorovic, and they gave postnatal day 7 rats a cocktail of agents. This included isoflurane, midazolam, and nitrous oxide. And they were able to show that there was extensive neurodegeneration in these rat pups. And it was not just accompanied by histopathological changes, but they also found learning and behavioral abnormalities in these offspring. And subsequent work was done in primates by Ansgar Brambring's group, and they've actually shown pretty much the same effects. However, we do not have a behavioral model for monkeys yet. So what is the public health importance of this topic? We know that millions of pregnant women and children do receive anesthesia, and if these findings were true in humans, this would create a big uproar. So we do have some data from the Mayo Clinic. It's data from Olmsted County in Minnesota, where they've looked at exposure to anesthesia during childhood, which they defined as less than 4 years of age, and the incidence of learning disabilities in the offspring. And I'm just going to give you a small snippet here. What they found was that more than 2 exposures within 4 years of age, or more than 2 hours of cumulative anesthesia exposure, was associated with a 2-fold increase in the incidence of learning disabilities and attention deficit disorder. These are 2 different manuscripts, but it just kind of gives you an idea that there is some association. However, these studies do not prove any causal association between these factors. And also, one important point to remember here is that the data comes from 1976 to 82. So the anesthetic agents used in those studies were completely different from what we deal with now. So there's no propofol, no isoflurane. It's all halothane, cyclopropane, and enflurane. So just keep that in mind when you interpret these studies. So let's move on to the various mechanisms that account for anesthetic neurotoxicity. Obviously, the one that's well studied is neurodegeneration. So developing neurons tend to migrate long distances, and they form extensive synaptic connections. So if you do general anesthesia at this particular phase in neurodevelopment, it silences these neurons, and the neurons that do not make active synaptic connections die out. So this is a programmed cell that it does happen spontaneously during development, but anesthetic seems to trigger and accelerate this process. And this is one of the most well-studied mechanisms of anesthetic neurotoxicity in this age group. But we also have several other mechanisms that contribute to anesthetic neurotoxicity. The other one that's actually reasonably well-studied, in rodents at least, is the impact of anesthetic agents on neurogenesis. And neurogenesis is generation of functional neurons from neural stem cells. And there are a lot of studies now showing that anesthetic agents, both inhalational and intravenous, such as Propofol, do decrease the generation of neurons, at least in vitro models. The third mechanism comes from studies by Laszlo Utskit's group in Switzerland, and they've actually looked at how anesthetics modulate spine formation in dendrites. And this is important because formation of dendritic spines is important for learning and memory formation. And they were able to show that all classes of anesthetic agents impaired dendritic spine formation, and given especially between the first 14 days of life in a rodent, which is equivalent to anywhere between the third trimester to up to 3 years of life in humans. And the final mechanism, which has kind of assumed more importance lately, is the impact of anesthetic agents on glial cells, especially oligodendrocytes. As anesthesiologists, we are always keen on neurons, but we need to remember that glial cells actually constitute more than 90% of the brain volume and plays important supportive roles in neurodevelopment. And it turns out that anesthetic agents, especially isoflurane, seems to cause oligodendrocyte death, especially when given during this period of vulnerability. So what we know so far, we can kind of summarize into three main categories. We know that anesthetics are neurotoxic, and this is a developmental age-dependent phenomenon. So if you give anesthetics to a middle-aged person, it's probably not going to do much. But if you give it during synaptogenesis, which is the early years of life, or later in life, there's considerable evidence to suggest that it does cause neurotoxicity. The second take-home message is that a combination of anesthetic agents is probably more neurotoxic than a single agent administration. Multiple studies have shown that if you mix a GABA mimetic agent with an NMDA receptor antagonist, the combination causes a much robust neurodegeneration than administration of a single agent alone. And I think that's a very clinically useful evidence to remember. And finally, it's the duration of exposure. And we do know that there's a dose-dependent phenomenon. Prolonged duration of exposure to anesthesia does cause more neurodegeneration, whereas if you give anesthesia only for a couple of hours, probably not as much. And in fact, the study which I recounted before from the Mayo Clinic showed that the effects of anesthesia are dose-dependent. So the big question is, most of the data comes from neonatal rat pups. So is this relevant in utero? So is the fetal brain really vulnerable to the adverse effects of anesthetic agents? And I think we are comparing apples with oranges here. And I'm going to tell you why the fetal brain is completely different from the neonatal brain. So the first step here is for you to understand the various stages of neurodevelopment. We start with neurogenesis, which I alluded to before, and then these newly formed neurons migrate really long distances and then differentiate, make synaptic connections, and then finally they remodel and establish new circuitry. So most of the studies are centered around this phase of synaptogenesis, which in humans is essentially a postnatal phenomenon. But what we are interested in here is the impact of anesthetic agents on the stages that precede synaptogenesis, which is neurogenesis and neuronal migration. And worryingly, most of these processes are actually modulated by GABA and glutamate, precisely the same neurotransmitter mechanisms that are modulated by anesthetic agents. So is it possible that we are putting the fetal brain under peril inadvertently by administering anesthetic agents to the mother? So the second major concept to understand is that the GABA is excitatory during development. So we all know that GABA is inhibitory neurotransmitter in adults, but in the developing neonatal period, it's actually excitatory. And the reason why it's excitatory is because one of the chloride channels that extrudes chloride away from the cell is there's a delayed development. And what it causes is accumulation of chloride in these cells. So when these cells are triggered by GABA, what happens is an exodus of chloride from the cell, causing depolarization rather than hyperpolarization, which is seen in mature cells. So this is a very important concept to remember. And the third major problem with GABA especially is that it plays critical roles in corticogenesis. It's essential for generation of neurons and also migration of interneurons. And we know that interneurons form the inhibitory circuitry in the human brain. And so if you accidentally impede or derail any of these processes, the consequences could be profound. The third major difference is the fetal brain is bathed with maternal hormones, and this phenomenon does not exist in the neonatal brain. We know that progesterone and estradiol especially increase up to 40-fold during pregnancy. They freely cross the placenta and change the chemical environment in the fetal brain. And we do know that progesterone does influence neural stem cell proliferation in the fetus and also interacts with GABA-A receptors. The same thing with estradiol. It alters several subcellular mechanisms. And the third main factor here is oxytocin. And you might be wondering how oxytocin, but it turns out that oxytocin does have GABA-A receptor modulating properties. And there's two well-published articles in Science showing that oxytocin transiently converts this excitatory action of GABA to inhibitory, especially at the time of childbirth. And the speculation here is that oxytocin somehow prepares the fetal neurons for a traumatic childbirth. From a completely philosophical perspective, you need to realize that GABA is a highly evolutionarily conserved molecule. It's actually essential for pollen synthesis. It is essential for ripening of tomatoes. It's not just mammals, so it's also really important in the plant kingdom. And also in the insect kingdom, it's essential for development of antennae in beetles and brain development in Drosophila. And so what is important here is that if you have an evolutionarily conserved molecule, the golden rule is please don't mess with it. So that's one of the key factors, because when I started looking into the literature, I was so surprised to find that GABA is so ubiquitous. And it's one of the commonly investigated molecules in the plant kingdom, and we have absolutely no idea about it. So far what we have is a tale of two trimesters. What do anesthetic agents do to the fetus? We do have a lot of data on the effects of anesthesia during this first trimester. We do have a lot of studies showing that anesthetic agents, especially when given as a single administration, do not cause an increase in the incidence of congenital birth anomalies. We also have data from the third trimester suggesting that anesthetics do have a transient effect on newborn parameters, especially change in umbilical cord gases and APGAR scores, but these are not clinically important. And moreover, we tend to use general anesthesia in babies that are compromised in utero. So there's kind of a confounding variable right there. And the big unknown here is the second trimester. And traditionally, it's been assumed that the second trimester is a safe period in terms of teratogenesis, understandably. But it also turns out that the second trimester is a period of bustling fetal brain development. Most of these processes, neurogenesis and neural migration, are happening at a frenetic pace. And it's possible that giving an anesthetic agent to the mother in the setting of, for example, non-obstetric surgery could change or derail some of these processes. So I'm just going to highlight some of the studies from the third trimester first, and then move on to the second trimester. So one of the first studies that specifically asked this question, if a single clinical exposure to 1 mg of isoflurane during the third trimester would impact fetal brain development. And this study was done at UPenn, and the authors kind of came to the conclusion that 1 mg exposure to isoflurane in the third trimester in rats did not cause any histological changes, nor were they associated with behavioral changes. And this study was expanded by another group, and they looked at dose-dependent effect here. And you can see here on the graph on the right of the screen, caspase-3 is a marker for programmed cell death. And you can see here that the higher the concentration, which is 3% isoflurane, you see a greater proportion of cell death, whereas this cell death was not noticed at the clinically relevant concentration of 1.3%. What's the evidence from human studies in the third trimester? Again, there's only one study published so far, which comes from the Olmsted County. And this study was a population-based birth cohort, again, from 1976 to 82. So they identified almost 5,000 infants born to mothers, out of which 4,800 were born vaginally. And among those that were born via cesarean delivery, there were about 500 of them, but 200 got general anesthesia, and about 300 got regional anesthesia. And then they tracked the development of the offspring in terms of educational achievement and learning disability. And this cumulative percentage of learning disabilities is now shown as the Kaplan-Meier estimate here. And you can see here that up to 8 years of age, it's really impossible to tease out any differences in any of the groups. So this is important because most of the studies, we look at short-term outcomes, but you can see here that up to 8 years, there's no way you could tell. But you can notice that after 10 years of age, the curves kind of gradually separate out. And you can see here that vaginal delivery, and irrespective of the type of anesthetic, is not associated with an increased incidence. The incidence is the same pretty much for vaginal deliveries as well as for cesarean deliveries under general anesthesia. The only interesting trend that authors noted here was if cesarean delivery was done under a regional technique, the incidence of learning disability was lower by 5%. And they speculated that it could be a decrease in catecholamines or other factors which have not been investigated so far. So is the second trimester vulnerable? Why is this question important? Again, a lot of non-obstetric surgeries, fetal cardiac interventions, and also, surgery, trauma surgery in pregnancy. A lot of surgeries are done in the second trimester, where the fetus is usually an innocent bystander. And these procedures usually take a really long time, usually 2 to 3 hours sometimes. And sometimes we do use a higher MAC to achieve uterine quiescence. So is it possible that we put all these babies at risk? So we created a rat model in our lab, and what we did was we exposed rats in the second trimester, which is gestational day 14, to one of two treatments, control and isoflurane 1.4% for 4 hours. And then we monitored hemodynamics and ensured that they were stable. And then we recovered these dams, put them back in the cages, and they delivered on day 22, 8 days after anesthesia exposure. And after delivery, we raised the offspring up to postnatal day 60, which is adulthood in rats. So just to recount, the anesthesia exposure happened in the second trimester, and we did behavioral testing in the offspring at day 60, which is adulthood. So it's only one exposure followed by testing really later on. And this is a scheme of what we did. We did locomotor activity, elevated plus maze, novel object recognition, and radial R maze. I'm not going to go into the details of all of these behavioral methods, but what we found was that exposure to isoflurane caused a profound impairment of spatial memory after in utero isoflurane exposure. So you can see here that rats that were exposed to anesthesia, shown here in red, took a really long time to complete a radial R maze, which is a hippocampally mediated task, suggesting that there's some impairment of hippocampal function. So it was not just the time taken to complete the maze, but also the number of omission errors they did on the maze. So it's not just one particular aspect of hippocampal function that was impaired, but multiple functions. So the question here is, what are the possible mechanisms behind these behavioral abnormalities? And we do have some data from other studies suggesting what could be going wrong in the fetal brain at this particular period of neurodevelopment. So the first study actually comes from UCSF, Jeff Saul and Greg Stratman's group, and they looked at the impact of isoflurane on neurogenesis in a cell culture model. And the first graph shows a fold increase in the number of cells. And you can see here that with exposure to isoflurane, there's a 42% decrease in the total number of cells at 48 hours. And the graphs on the right actually show the percentage of proliferating cells. BRDO is a marker for proliferation. And you can see here that administration of isoflurane causes a profound decrease in the number of proliferating cells. And they also noticed that this decrease was not transient. So when they repeated the experiments 12 hours after exposure, it was still shown to impair proliferation. So it's not just a transient effect. It seems to be a long-lasting effect. And again, we in our lab asked if this is a dose-dependent phenomenon. So Jeff Saul's study actually used 3.4% of isoflurane, which is a really high dose if you think about it. So we asked if using lower doses of isoflurane would cause this phenomenon. And we found out that if you use 0.7 or 1.4%, we did not find any changes in proliferation of these neural stem cells. But at 2.8% isoflurane, we did see an impairment of proliferation by 30%, and it lasted for up to 24 hours after exposure. So it seems to be a dose-dependent phenomenon. So the second study, which I'm going to highlight, is the possibility that administration of anesthetics per se could induce inflammation in the fetal brain. Now, this is a very unique concept. How could anesthesia induce inflammation? So this study came from Massachusetts General Hospital, where they exposed, again, pregnant mice at embryonic day 14, which is equivalent to the second trimester. And they harvested the fetal brains to study markers of inflammation. And you can see here that exposure to sevoflurane, shown here in the blue columns, was associated with an increase in interleukin-6 levels, and also an increase in caspase-3 activation, which is a marker for programmed cell death. On the other hand, exposure to sevoflurane was associated with a decrease in synaptophycins and a decrease in postsynaptic density markers, which are markers for active synapse formation. So it seems that exposure to sevoflurane just for 2 hours during the second trimester causes a downregulation of synapse formation and an upregulation of inflammation in the fetal brain. What about the monkey models? And again, this study comes from Ansgar Bramping's group, and they picked ketamine as their agent of choice. And they did this because it was easy to administer, and they already had a working model. And so what they did was they exposed 2 different groups of monkeys, pregnant macaques as well as neonatal macaques. And the idea here was to see if the fetal brain was more vulnerable to ketamine neurotoxicity. And the primary outcome measure in the study was neuroapoptosis in different brain regions. And this here shows the computer plot depicting the patterns of cell death in ketamine-exposed versus control fetal monkey brains. The black dots represent cell death profiles, and you can see here that in utero exposure to ketamine enhances apoptosis, especially in the frontal cortex, amygdala, thalamus, and the cingulate cortex. But what was more striking about the study was that it raised the possibility that the fetal brain is perhaps more vulnerable than the neonatal brain to anesthetic neurotoxicity. And in this graph, you can see the mean number of dead neurons along the vertical axis and the developmental age along the horizontal axis. And you can see here that ketamine, which is marked in blue, causes more apoptosis in the fetal than in the neonatal brain. In fact, the number of neurons that were affected by ketamine exposure in the fetal period was almost twofold higher compared to the neonatal period. And this group also extended the study with Propofol, a similar methodology. They used gestational day 120, which is equivalent to the human second trimester, neonatal macaques, and an IV Propofol infusion in a very standardized setting. And they quantified neuroapoptosis in multiple brain regions at 3 hours. Similar findings here. You can see here that AC3 is activated caspase-3, which is a marker for cell death. And again, Propofol in the fetus was associated with a 2.5-fold increase in cell death, whereas it caused a much greater degree of apoptosis in the neonatal period. And finally, they repeated the experiments with isoflurane, and again, the same methodology, and they gave isoflurane for 5 hours this time. And the findings were pretty much similar to both ketamine and Propofol. But what was interesting about this isoflurane study is they also quantified degeneration of oligodendrocytes in addition to neurons. And the degenerating oligodendrocytes are shown here as red dots. And they were actually able to show that oligodendrocyte degeneration was 4-fold higher after exposure to isoflurane. So it's not just neurons, but oligodendrocytes die as well. So let's kind of recap all the possible mechanisms that could be contributing. So we have inhibition of neurogenesis. We have permanent alteration of synaptic mechanisms. This was a wonderful study from Mass General Hospital. And then we did see induction of inflammation in the fetal brain, as shown by an increase in the interleukin-6 levels after exposure to anesthesia. And finally, we do have evidence to suggest that the second trimester fetal brain can respond to anesthetic exposure by neuronal and oligoapoptosis. What about the use of Propofol? I did highlight one study in the monkeys with Propofol. The safety profile is still unclear. We do not have any behavioral studies with Propofol. And if you recollect, Propofol seems to be associated with a marginally lower degree of apoptosis compared to ketamine and isoflurane. So it's possible that Propofol could be an alternative, but unless we have good behavioral studies, it'll be hard to come to that conclusion. So what are the caveats? This is really important. I was a big skeptic when I started doing this research. In fact, when I started, I said that the fetal brain was not vulnerable to anesthesia because it's in a protected environment. And my findings turned out to be the opposite, and then I started to buy into the whole topic. But there are a lot of caveats. If you read the literature, you need to read that with a pinch of salt. Okay, so one of the first main problems that pops up with these studies is how do you extrapolate results from animal studies to humans? And this is very important because both rats and humans are different when it comes to the neurodevelopment. What stage in rats compares to what stage in humans? We really have no idea. But we do have one tool. It's on the internet. It's called translatingtime.net. And in case if you're interested, you can actually go in and plug in numbers, and it converts developmental age in rats in any different type of animal model and converts it to the human equivalent. Again, it's a very simplistic tool, and you know that the brain, different parts of the brain develop at different rates, so it's really hard to make any meaningful conclusions from this conversion. But I think one of the biggest criticisms, which is a reasonably fair criticism, is the longer relative duration of exposure to anesthesia. For example, in the rat model, which I told you before, the total period of rat gestation is 22 days. And if you expose these rats to 4 hours of anesthesia, it's equivalent to about 48 hours of anesthesia in humans. So, this is a very simplistic mathematical calculation. And so this criticism is appropriately valid, but there are a lot of other things which you need to take into account. The human brain is much more complicated than the rat brain. And you can see here that the number of neurons in the human brain is almost 86,000 billion. And you can see that the cortical size is almost 82% compared to only 42% in rats. And neurogenesis cycles, which actually refers to the number of rounds of proliferation that one neural stem cell goes through, is actually much, much higher in humans. So if the insult is severe enough early in development, you can have a lot of downstream consequences. And also the migrating distance is much higher in human brains compared to the rodent brains. So, you have to take this criticism with a pinch of salt. And the other criticism is, what about hemodynamics instability? If you look at the older studies, almost 25% of neonatal rat pups died. So the mortality rate with these studies is 25%. It never happens in human clinical anesthesia. So most of these rat pups developed hypercarbia and hypoxia. So is it possible that it's all these metabolic derangements that are causing the apoptosis and not the anesthesia? But this criticism has been addressed by Vesna, where she used a guinea pig model, which was easy to instrument, and therefore they were able to do invasive arterial pressure monitoring and also check glucose and a lot of other metabolic parameters. And they were able to show that even with good control of hemodynamics, they were able to show neurodegeneration in the fetal brain. So it's not just impaired hypoxia and hypercarbia, but also it's possible that anesthesia alone can cause this problem. So I'm going to end the talk with this slide. This is kind of the take-home message. What will you tell the mother? You come across a patient who asks, I read somewhere that anesthetics cause neurotoxicity. Well, you can say that, if it's patients in the first trimester, you can definitely tell them that there's no evidence whatsoever in the clinical literature to suggest that anesthetic exposure causes any significant increase in congenital anomalies. And for the second trimester, you can tell them that there's lack of human data, though there's some emerging animal evidence suggesting that this could be a possibility. And especially during the third trimester, it has no adverse effects whatsoever. Okay, I'd like to conclude my talk here, and I'll be able to take questions later on. Okay, thank you.